know the scope of the problem of neovascular ocular disease. Even with the advent of anti-VEGF treatment, as a clinician, I can tell you that there are still a majority of our patients who present with poor vision, and despite aggressive treatment with frequent injections into their eye and frequent office visits, still remain with poor vision. Towards this end, our company's objective has been to try to characterize more stable synthetic molecules that can be given by dose by both an induction treatment and potentially sustained release treatment. Carboxyamid triazole is one of these molecules. It was initially characterized at the National Cancer Institute extensively. Small molecular weight compound. It's a potent anti-angiogenic preclinically and has been in clinical trials of almost 900 patients with a good safety profile. It blocks capacitive calcium entry and intracellular calcium via trap channel and antagonizes many VEGF-mediated events. In clinical trials, efficacy was limited, but these trials were directed at advanced metastatic disease, so clinical as a monotherapy, so clinical trial endpoints were hard to achieve. It's also highly plasma protein bound. We felt like in the low protein environment of the vitreous, this compound would perform quite nicely. I don't know if we have up here. That was our prevention study. I can talk more about that on the next slide. But we looked at regression. Here are animals treated with vehicle control and an oxygen-induced retinopathy model. And with CAI, there's significant reduction of neovascularization, and the neovascularization appeared fibrotic and bud-like, which is what we tried to achieve in our clinical treatment. If we actually look at quantitate this effect in this model, we had quite a robust inhibition of neovascular cell nuclei. It was the most potent molecule that was characterized in this model to date. So we then sought to do an intravitreal formulation. The other thing that we found in this model was it upregulated BCL2, rescued normal histopathology, and decreased tunnel and DNA nicking, consistent with imparting neuroprotection. So with Gideon Shapiro, my colleague, we did an intravitreal formulation. Our first intravitreal formulation was the Cyclodextrin Complex, which we felt would give us some area under curve or sustained release dosing. Here in collaboration with Maria Grant, we showed that it had a robust inhibition in a laser CNVM model of choroidal neovascularization. And we then sought to do safety testing. There are no effects on retinal photography or fundus imaging. And we can see that there is molecule still present at 2 months. No toxicity was observed in histopathologic studies, VEPs, or ERGs. We felt that with the cyclodextrin formulation, we had some sustained release, but there were some issues with sterilization, so we moved to a nanoparticle formulation. And with the nanoparticle formulation, we were able to, with Carl Beckett at Caveris, achieve a stable nanoparticle incorporation. It's easily filter sterilizable. It can be administered with a 30-gauge needle. And similar to the cyclodextrin formulation, there's no toxicity observed with respect to exam, IOP, histopath, or chemistry. If we look at the efficacy of the nano formulation, similar to the cyclodextrin, it's anti-proliferative in cell lines, and with the laser CNVM model, here we've done with Maria Grant, shows that it has a fairly robust inhibition. We did not see any synergy with anti-VEGF, but these are early experiments. About 6 months ago, Carl came to me and said, well, we think we can get this into PLGA. Would that be something good? And I said, well, if we can get a sustained release anti-angiogenic that's safe, that would be great. And indeed, recent experiments show that we're able to stably incorporate this molecule into PLGA. We're able to get it in with efficiency similar to dexamethasone, so that we feel that we are able to engineer CAI into a PLGA matrix with a sustained release comparable to the already approved dexamethasone Osherdex implant. So in summary, we have been able to characterize a potent anti-angiogenic molecule. It's found to be safe in two different intravitreal formulations. It's got a great safety profile in systemic administration in human disease, and that this molecule can be designed for both induction treatment as well as sustained release. I'd like to thank my collaborators, and thank you.